Peace be upon you, grade 8 students. In this video, you are going to study each of these objectives. In this video, we are going to see an application about the secondary immune response and its importance. Let us make revision. We have said that the specific immune response is primary if it occurs after the first contact with an antigen, while it is secondary if it occurs after the second contact or more contacts with the same antigen. The secondary immune response is fast because it occurs directly by the memory cells, efficient because it gives a big number of effector T or B cells, and durable or long-lasting because it lasts for a longer time. Previously, many children died because of a disease called the smallpox, in addition to other diseases such as tetanus, hepatitis. But now these diseases do not cause death in children because scientists tried to find the solution. They worked hard and hard until they found it. The solution for this problem was by vaccination. A microbe can either be virulent or attenuated. Since we are going to use these terms, please pause the video and take notes. A virulent microbe is a harmful microbe that causes disease, while an attenuated microbe is a weakened microbe that does not cause disease. Look at the armies in these pictures. What do armies make? Yes, armies make maneuver before the real wars to become ready for them. Do you know that the same thing can be done in our bodies and that our immune system makes maneuver? Vaccination helps our immune system making maneuver, and this is all happens without being damaged or sick. Let us see how. How can people be protected from the infection by a virulent pathogen forever? What do you think with bringing the virulent microbe and making it weak, then exposing the person to it? What is the effect of this action? Think well. The effect of this action is that the immune system of the person makes an immune response against the weakened microbe and produces memory cells for it. So can we apply this thing? Yes, because being exposed to the weakened microbe is like doing a maneuver before being exposed to the virulent one. So people can be protected from infection by a virulent pathogen forever by the administration of attenuated pathogens into their bodies. Keep in mind, administration means taking, and attenuated means weakened, so taking the weakened pathogen helps the person from being infected by the virulent pathogen forever. Some of the childhood diseases that are caused by virulent pathogens and resulted in death of millions of children are polio, hepatitis, rubella, and tetanus. All of these diseases are very dangerous and can be prevented or be with reduced symptoms by being vaccinated against them. For example, how can we prevent polio disease in children? We can prevent polio disease in children by administration of attenuated polio virus in them so that they will not get the polio disease if they are infected by the virulent polio virus. Similarly, how can we prevent rubella in children? We can prevent rubella in children by administration of attenuated rubella virus in these children so that they will not get the rubella disease if they are infected with the virulent rubella virus. So, these diseases can be prevented by being vaccinated against them, which means by the administration of the attenuated microbe. Let us define vaccination. Vaccination is the administration of attenuated or killed microbe or any of its substances into the body. Administration means taking. Attenuated or killed microbe is called the vaccine. A vaccine can be administered or given either orally or by injection. Orally means in the form of drops given in the mouth. How will your immune system react when you are vaccinated against a disease? Yes, this vaccine, which is an attenuated microbe, induces a primary specific immune response with the formation of memory cells. What is the role of these memory cells? Memory cells will fight directly and quickly the virulent microbe when it enters the body. Does the vaccine help you develop an immunity against a virulent microbe? Are you in danger anymore? Of course not. You will not become in danger anymore because the vaccine helps you to develop an immunity against the virulent microbe. To study the role of vaccination, a scientist performed an experiment on chicken where he used attenuated and virulent cholera bacteria. What is cholera? Cholera is a lethal or deadly disease caused by cholera bacteria. Let us read this experiment very well. Look at this legend. What does this legend mean? It means injection. So once you see this drawing, you should use the word injection or the term injected with. Chicken A is injected with virulent cholera bacteria, and after 1 to 2 days, chicken A dies. Chicken B is injected with attenuated cholera bacteria. Then after 7 days, it is injected with the virulent cholera bacteria. Then chicken B survives. We want to analyze this experiment. Remember that when you are asked to analyze, you should mention the results and the conditions. Let us take chicken A. What is the result? Chicken A dies. What is the condition? The condition is injection with virulent cholera bacteria. So we say chicken A dies after 1 to 2 days when it is injected with the virulent cholera bacteria. What about chicken B? Do we have different result or same result? We have different result, which is the chicken B survives. We should write while chicken B survives. What is the condition of chicken B? Is that it's injected with the attenuated cholera bacteria, then the virulent one after 7 days. So you write while chicken B survives when it is injected with attenuated cholera bacteria, then with the virulent one after 7 days. Conclude the role of vaccination. To conclude, you should study the effect of the different variable. What is the reason behind having different results in chicken A and B? It is that chicken A is injected directly with virulent cholera bacteria, while chicken B is injected with attenuated cholera bacteria before the virulent one. Remember that the injection of attenuated cholera bacteria represents vaccination. So chicken B is vaccinated against cholera while chicken A is not. Does chicken B get an immunity against cholera? Yes, because it survives. Does it become sick with cholera? No, it doesn't. So vaccination induces a developing immunity and prevents disease. This graph shows the variation of the amount of anti-cholera antibodies in the blood of chicken B, which is measured during the 2 injections. Look, the injection of attenuated cholera induces a primary specific immune response, which is slow to start and not very efficient, but with the formation of memory cells. Then the injection of virulent cholera induces a secondary specific immune response, which is fast and efficient because the memory cells are activated and will start producing antibodies in high amounts. So what prevents the cholera disease in chicken B? Why does it get immunity against cholera disease? Because the antibodies produced upon the injection of virulent cholera will eliminate it quickly before it causes disease. What can you say about the role of vaccination? Vaccination strengthens the immune system against virulent microbes, which means that vaccination makes your immune system stronger to fight a virulent microbe. Vaccination prevents sickness with infectious diseases. Also, vaccination protects against infectious diseases. Please, dear students, pause the video and take these notes. How does vaccination work? What happens when the virulent microbe enters the body of a person vaccinated against it? Pay attention. Vaccinated means given an injection of the attenuated microbe. Upon the injection of the attenuated microbe, which is a vaccine, a primary immune response is induced with the formation of memory cells. So upon the entrance of virulent microbe, the memory cells are directly activated and quickly produce antibodies. These antibodies will bind to the antigens of the virulent microbe and neutralize it before you become sick. A vaccine is given usually in more than one dose. This graph shows the variation of the immune response induced after taking 3 doses of a vaccine with respect to time. Look, with every dose, the immune response becomes stronger. Why? Because with every dose, more memory cells are produced, and these memory cells, which are built by the repetition of the doses of the vaccine, can last for more than 20 years or for a lifetime. As a conclusion, we can say that with every vaccine dose or injection, the immune response gets stronger and produces more memory cells, B and T lymphocytes. What is the importance of repetition of vaccination? It's important to take the same vaccine but in several doses in order to develop complete immunity against the virulent pathogen. Note that it is also an acquired immunity because the immune system of the vaccinated person will be working and fighting the virulent pathogen by a secondary immune response. And in order to allow the immune response to last longer and to develop enough antibodies to fight the virulent pathogen, this is all will lead to highest protection against the virulent microbe. Here is a vaccination schedule. The vaccination schedule includes all the necessary vaccines that a child should take during his childhood. As we have said before, it is important to take the same vaccine but in several doses and in particular at specific ages. For example, the vaccine against polio. The vaccine against polio is taken at ages 2 months, 4 months, 6 months, and from 4 to 6 years old. Suppose a person is vaccinated against a certain microbe. He is injected with a toxoid. The toxoid is an inactivated toxin used as a vaccine. Inactivated means not active. His immune system develops a primary antibody response which is slow to start and characterized by a low amount of antibodies that disappear after a few days. This person is naturally infected with the same microbe but the virulent one, and this virulent microbe produces a toxin which is a toxic substance. Then his immune system develops a secondary antibody response which is fast and characterized by high amount of antibodies that last for a longer time. What are the characteristics of this acquired immunity? Look at the antibody and the antigen. Are they complementary? What does the high amount of antibodies mean? What do the remaining for a longer time indicate? We can deduce the following characteristics of the immunity provided by vaccination. This immunity is specific because the antibodies are specific to the antigen, and efficient because of the production of higher amount of antibodies against the virulent pathogen, and long-lasting because the immunity is for a long time. Please, dear students, pause the video and try to answer these questions. Choose the correct answer. Vaccination is a preventive method or curative method. A preventive method is the method used to avoid and prevent a disease, while a curative method is a method used to cure or treat a disease. The answer is vaccination is a preventive method because it is used to avoid a disease. Vaccination is applied before infection with virulent microbe or after infection with virulent microbe. What is a prophylaxis method? A prophylaxis method is the method used before infection with virulent microbe, while a therapeutic method is a method used after infection with virulent microbe. Think well, dear students. Excellent. Vaccination is applied before infection with virulent microbe, so it is a prophylaxis method. Dear students, please memorize the definition of preventive, curative, prophylaxis, and therapeutic methods. Its application time. Indicate the true statements and correct the false one or ones. Please pause the video after each sentence and try to give the correct answer. A vaccine is introduced into an individual's body to cure a disease. False. It is introduced into an individual's body to prevent or avoid a disease because vaccination is a preventive method. The administration of vaccine triggers the production of memory cells. It is true because it induces a primary specific immune response. In a vaccinated individual, after the exposure to the same pathogen, but the virulent one, memory cells are activated and start a primary immune response. It's false. They will start a secondary immune response. Application 2. In this application, you have to compare the immune response that takes place in an individual when vaccinated against a certain microbe and the immune response that takes place in the same vaccinated individual but when infected with the virulent microbe. The immune response that occurs due to vaccination is primary, slow, less efficient with less amount of antibodies, and the immune response that takes place in this vaccinated individual after infection with the virulent microbe will become secondary immune response, which is rapid, more efficient, with more amount of antibodies. Application 3. Choose among the graphs the one that shows the variation of the quantity of anti-cholera antibodies in the plasma of a chicken that received 3 injections of attenuated microbe. Please, dear students, pause the video, read and analyze the graphs very well, then choose the correct graph. Excellent. The correct graph is graph B. Let us see what is wrong in graphs A and C. In graph A, the quantity of anti-cholera antibodies decreases after the second and the third injections, which is wrong because the secondary immune response should be long-lasting. While in graph C, the quantity of anti-cholera antibodies produced after the first injection is more than that produced after the second and the third injection, which is also wrong because the secondary immune response should be more efficient than the primary one. At the end, thank you for your attention.